Thank you. Thank you very much for having me. Thank you also very much for having me speaking in English to you. If I start to go too fast or if I start to use English phrases that don't make sense when you translate them in your heads, please stop me. I'm happy to explain. I don't want to leave anybody behind. So, to begin with, I just want to make it clear that humans have had a long history of modifying animals, and I know you've been talking about this already in this meeting. All of these here are examples of modifying animals, even training animals. When you train an animal, you modify them and you change them. And some of these modifications here you can see result in pain, suffering and distress to the animal, and there's no gene editing or no gene alteration involved here. But is direct gene altering or editing different? I'd be really interested at the end of this talk to see whether you think it's different ethically or whether it's just another way of changing animals. So I'm going to begin by talking about genetically altered laboratory animals. So why are they created? Why would you want to do that? Well, they're mainly mice and zebrafish. Those are the two most altered species. And it's done for a number of reasons. Often it's done to create models of genetic diseases and disorders that affect humans, like cystic fibrosis and cancer. So if the disease is caused by a faulty genetic process in humans, then you can recreate that in animals and try to find a cure. It's also done to understand how certain genes function. So if you stop a gene from functioning and see what happens to the animal, then you've gained an understanding. But also increasingly, gene editing and alteration are spilling out of the laboratory, and it's proposed that animals outside the laboratory, like wild and farm animals, could also be genetically altered. And I think this is quite a step shift in the way that humans treat and use animals. So I'm going to try and avoid using too much jargon. There are a couple of terms that I just want to define because I know you're a multi-stakeholder audience. So the first term there, genotype. That's the animal's complete set of genes as stored in their DNA, the double helix that you've all seen. So that's the genotype. The phenotype is the animal as they actually appear. So it's their appearance and their physical properties as you see them. So obviously, the phenotype will be the result of an interaction between the genotype and the environment that the animal's brought up in. So when in this talk I refer to adverse phenotypes or harmful phenotypes, that means that the animal as you see them will be suffering as a result of the genetic modification that's been done to them. Now, before I go on to talk about the welfare impacts of gene altering, I just want to sort of go off on a sidetrack to explain how animal experiments are regulated, because it's important to set the context for gene editing lab animals. So, throughout Europe, animal experiments are regulated by a directive called 2010/63 EU. Each European member state has to transcribe this directive into its own national legislation, and that's your legislation there, the Finnish Act on the Protection of Animals used for scientific or educational purposes. So in the UK, we have the Animal Scientific Procedures Act 1986. And all of these national legislations have these fundamental principles in common. So you have to use a non-animal method wherever one is available. So if you want to do an animal experiment and there's an alternative, you're not allowed to do that experiment. Scientists have to use the minimum number of animals that they possibly can while still answering the scientific question, because obviously if you don't use enough animals, then your science won't be sound and those animals will have been used for nothing. And any pain, suffering, distress or lasting harm has to be minimised. So those principles there are also referred to as the 3 Rs: replacement, reduction and refinement. The refinement bit is all about reducing suffering and improving welfare. And this is the number of animals that are used in the European Union plus Norway. So this is data from 2018. This is the most recent data. It's come from this database called Allures. It's really easy to find and it's completely open access. So if you're interested in how animals are used across Europe, you can look that up. So you can see In 2018, there were 10.8 million scientific procedures using animals, mostly mice and rats. The other fish there, a lot of that is Norway, because they do a lot of experiments on salmon, sort of veterinary experiments on salmon because of all the salmon farms they have. So actually throughout the rest of Europe, you're talking about 90% of animal use is mice and rats. And in Finland, you used almost 120,000 animals in 2020. And I just want to tell you about a couple of other important aspects of the law before I move on. So there has to be a license required for every establishment that uses or breeds or supplies laboratory animals. Every science project using animals has to have a license, and every person doing experiments also has to have a license. And the project license is really important because it sets out exactly what will be done to the animals, how much suffering is caused, and it limits the amount of suffering that's going to be caused in each project. And there are also specific purposes for which animals are allowed to be used, like medical research, safety testing, that kind of thing. And in Finland, every project is reviewed by a project authorization board, and there's a URL at the bottom that leads to your legislation. I'm happy for everybody to have my slides after the presentation as well, so you can look these things up. And what's critically important about the project license is that it involves a harm-benefit analysis. So this is where the predicted harms to the animals have to be weighed against the likely benefits to humans, animals, or the environment, and the research has to be deemed justified before it can go ahead. So thinking again about genetically altered animals, project licenses are needed to create every new line of genetically altered animals for reasons which I'll come to. Licenses are also required just to breed genetically altered animals if they have a harmful phenotype. And of course, a license is required if a genetically altered animal is then going to be used in experiments that may cause pain, suffering, distress, or lasting harm. Because often being born or created as a genetically altered animal is just the start of the story. Often they'll go on to be used in further procedures, for example, to test a therapy for a human condition. So that's the sort of essentials of the law. And now I'll go back to talking about genetic alteration in the laboratory. So it all began with spontaneous mutations. Obviously, any kind of animal or plant, come to that, can have a spontaneous mutation in their genetic material that can lead to a disease or condition. So you've got the nude mouse on the top right there. They've got a very severely compromised immune system, so they're used in a lot of research. They were a natural mutant. There you've also got the MDX mouse, which spontaneously gets Duchenne muscular dystrophy. So it's deficient in an important gene called dystrophin, which creates a protein that strengthens human muscle cells. And so without that protein, the muscle cells are very weak and degenerate. Now, the mouse doesn't get Duchenne muscular dystrophy as badly as the human there, but they still suffer from the condition. And this occurred completely spontaneously. And when it was realized that they were a model of Duchenne muscular dystrophy, they continued to be bred, and they're still used to this day. So spontaneous mutations obviously are relatively rare, and they're also unpredictable and uncontrolled. So it's really just a sort of a opportunistic way of making the most of spontaneous mutations. So the next step was to create genetic mutations. And initially, this was done using either radiation or chemical mutagens. So a mutagen is a substance that causes DNA to mutate or change. All of these kinds of substances and conditions like radiation are obviously things that we avoid at all costs, and there are regulations that prevent us from coming into contact with them. But here, as you can see, animals are exposed to these mutagens. So this enabled researchers to hugely increase the number of mutations that occurred, but they were unpredictable. You can't control what they are. And you can see from these diagrams here, often the whole process is sort of cleaned up or sanitized. We see a lot of little cartoons like this to show how mutant animals are created. But that's not really how it is. These procedures involve causing animals pain, suffering, and distress. So this is an example of a protocol for mutagenesis in mice. So you've got 6-week-old male mice being injected with the mutagen into their abdominal cavity, and this makes them very sick, and it makes them completely infertile for a while, and when they begin to produce sperm again, these sperm are mutated. So obviously, as I've said, the mutagens are dangerous. It can be fatal to the animals, and animals can also get cancer from these mutagens. They're mated with females to see when they become fertile again, and then when the females begin to get pregnant, then the mutant lines of mice are created. This happens on an enormous scale, so this still happens now. We're talking thousands of animals who are used in these kinds of procedures, and the mutant phenotypes themselves can be quite severe. And then obviously, once a mutated line has been created, researchers need to see what the animal's phenotype is. They need to characterize the animal. So this is just a diagram showing some of the so-called phenotype screens that are done to animals to understand what the mutant is. The one on the top right there with the wheels, that's called a rotarod test, and that slowly turns round, and you see whether the animals are able to stay on or whether they fall off onto a padded surface below. The little footprints represent gait analysis to see how the animals move. You can see there's a swim test there. The bottom right is a test to startle them and to see what happens. So all of these are also likely to cause stress. You know, each one on its own may not be that stressful, but when you put them all together, it can be significant. I said I'd mention zebrafish too. So this is an example protocol for mutagenesis in zebrafish. So they're actually immersed in a solution of this ENU, this mutagen. And as you can imagine, they cannot get away from this. So it's going to be pretty distressing, and that can also happen to them more than once. As with the mice, the mutagen causes suffering. There's a risk of harmful phenotypes, and there's also a lot of mortality and wastage. Those diagrams there are just showing you the sort of phenotype testing that's done with zebrafish, so they're sort of video tracked to see how they move and how they behave. So the next step then, we're sort of increasing in precision. The next step is genetic alteration using recombinant DNA technology. So recombinant refers to the act of combining DNA from different species. This is where scientists became able to create transgenic animals, so putting genetic sequences from one species into a completely different species. So the first transgenic mouse was created in 1974, and the first transgenic zebrafish about 10 years later. And initially this was done by directly inserting genes into a fertilized egg in vitro or in cell culture. And you can see a picture of it occurring there. So these needles literally go away to nothing when you look at them. It's an extremely painstaking and precise process. It's called pronuclear injection. Initially, the success rate of this was around 3 to 5%. So again, a lot of animal wastage. The next step was to alter the genes in cell culture before they're put into the fertilized egg. That didn't increase the success rate particularly much, but the final step, or the subsequent step that helped this to become more accurate, was using a viral vector, which is a piece of DNA material from a bacteria that's used to take the material that you're trying to introduce into the fertilized egg. And that has increased accuracy considerably. So now you hear success rates of between 80 and 100% when viral vectors are used. And this is how researchers are able to more accurately knock genes out, to deactivate a gene to find out what happens when the gene isn't activated, and also to knock genes in, and this is where the transgenes are put into the animal. So that's all the sort of technical aspect, but obviously we're here to think about animal welfare. And there are painful and distressing procedures that need to be done in order to generate these animals. So if you want to genetically alter a mammal, then there are procedures that are needed to source the eggs. You need to be able to access an egg. So obviously, to create genetically altered fish or birds, they produce eggs externally. If you want an egg, there's your egg. For mammals, you have to inject female mice, for example, with hormones to make them super ovulate to generate more eggs. And the females are then killed for their eggs. The eggs are genetically altered, And then the procedures have to be done to prepare another female to receive the egg as a pregnancy, which also involves hormone injections, and then embryo transfer, which can be surgical or non-surgical. So the picture on the top there, sorry, the colour's a bit strange. It was very dark. I had to sort of highlight it. I don't know if you can see there's a scar on the mouse's back there, and that's where she's had a surgical process to implant the embryos. The mouse below there, she's under general anaesthetic, and you can see she's having a non-surgical embryo transfer. So procedures are involved to generate these animals. The phenotypes are still not always predictable, and there is still wastage involved. And there are welfare issues for the genetically altered offspring as well. There's the phenotyping procedures that I mentioned earlier, and also they have to be genotyped. So you need to take a piece of tissue to analyse the DNA and to see whether you've got the alterations that you wanted. In the case of mice, this always used to be done by taking a piece of tail from the end of a young mouse, and it was thought, well, this is just a tiny piece of tissue, it won't hurt them. But when somebody actually looked with a microscope, there are nerves going all the way to the end of the tail tip in a juvenile mouse. And the researchers also observed fragments of bone and neuromas, like knots of nerve tissue that are associated with phantom limb pain in humans. Some progress has been made, so often now if mice are identified using ear notches, so the ears are notched in different places so that you can tell individuals apart, it's possible to use that tissue for DNA analysis now. Zebrafish are often genotyped by clipping their fins, which is also painful. You can see differences in behaviour if they have pain relief or not. And of course the phenotypes themselves can be harmful. Just on the zebrafish fin clip and the mouse genotyping, it's possible to use a cheek swab and also to swab the side of the fish to get DNA there too. This isn't being done often enough, so this is a campaign that my organisation has set up to get people to stop fin clipping zebrafish. And now the most accurate method for gene editing animals that you've probably heard of in the media is CRISPR-Cas9. This is a newer technique. And this uses a bacterial gene sequence, like the other techniques, to guide in the DNA that you want to introduce. And the Cas9 part of it is an enzyme that cuts through the DNA. So this picture shows a double strand of DNA that's been opened up. The Cas9 goes in and cuts through the DNA, and then the CRISPR guides in the sequence of DNA that you want to introduce. So this has increased accuracy, and again you see lots of little cartoons like this showing what happens to the animals, but it's also led to an explosion in animal gene editing, which I think is of a concern, despite the fact that yes, experiments using gene-edited animals can lead to medical progress, they can lead to greater understanding, but I don't think it makes it right to do anything to these animals and to cause this huge expansion in gene editing. So any scientific use of animals, gene alteration included, raises welfare, scientific and ethical issues. So as we've just discussed, the welfare issues associated with gene editing include these painful procedures to generate the founder animals, genotyping and phenotyping, the harmful phenotypes, especially if they're disease models, and the phenotypes can be unpredictable, they can be unstable, so it's really important to make sure that animal welfare is properly assessed as well as the phenotypes, and they're often used in other scientific procedures too. So this is to show you how many or the proportion of genetically altered animals that are used now. So this is from the Allures database again. You can see around 3/4 of animals used in Europe aren't genetically altered, so that's about 8.3 million animals there. The blue is animals who are genetically altered, but the phenotype isn't harmful. That's about 2.1 million animals. And the black slice there is where animals are genetically altered and they do have a harmful phenotype. So that's about 400,000 animals a year. Looking at the ones that do have a harmful phenotype, you can see here the numbers of animals who are experiencing mild, moderate, and severe severity. That's how the severity is classified. So about 57.5% of genetically altered animals with a harmful phenotype are experiencing moderate or severe severity. So that's about 226,000 animals a year. Of the animals who are experiencing severe suffering, as you can see, mostly mice, rats, and zebrafish, and a couple of pigs. So those are the welfare issues which, although it's a small percentage of animal use overall, that's a large number of animals, and obviously every individual animal is important. And there are ethical issues too, including the huge wastage of animals' lives. Even if they don't suffer, I think death is a harm to animals. All animal lives are important. Gene editing can make instant and significant and multiple changes to the genome. So it's actually directly editing what's been referred to as the blueprint of life. Genes from different species can be added, which is also something completely new. And as I'll come to at the end of this talk, I think gene editing techniques are being applied to non-laboratory animals without proper public understanding or consent. It's racing ahead of people's consent to do this. And a technique that involves intellectual rights, I think, is also very different from conventional breeding. So for the scientific issues then, first off, I want to clarify that everything is a model. So I'll come to the grade bits of this slide in a moment, but I just want to explain what I mean about everything being a model. So all of these techniques here are used in research and testing. So for example, when any drug is developed, it won't go straight from animals into humans. There's a whole process here. And you can see, so on the top left, that's a computer prediction of how a particular potential drug will behave. Next down there, you've got cell culture, where the person's sort of injecting something into a flask. The top middle shows an organ on a chip, which is a relatively new, complex, three-dimensional cell structure that actually mimics an organ on a chip. So, for example, there's a lung on a chip that will exchange gas and that you can actually make breathe. Invertebrates like the fruit fly are also used. Primates are used as models for humans. Conventional and genetically altered animals are used as models for humans, and humans are used as models for other humans too. That picture there shows an Israeli soldier who's part of a clinical trial for the COVID vaccine. All of those techniques were used to produce the COVID vaccine, including human clinical trials. Human clinical trials are done according to guidelines called the Declaration of Helsinki, which is one of your gifts to the world. After the Second World War, this was set up in order to protect people from abuse in experiments. So everything is a model. I'd be another model for a middle-aged woman. You know, nobody can actually tell what a drug will do until you take it. So genetically altered animals are another model. Sometimes they can be a more accurate model. If they've been altered so that they precisely reproduce a particular pathway in a human disease, then they can be more accurate. But there's still a translatability or reproducibility crisis going on in science at the moment, where experiments either can't be repeated or the model fails. And there are a number of reasons for this. Experimental design is not good. I could give you a whole lecture on that. It's partly due to scientific culture and the pressure that scientists are under to generate more results, perhaps by losing attention to detail. Reporting could be improved. There are a lot of initiatives going on at the moment to improve experimental reporting. And also experimental confounds aren't always recognised. So, for example, a lot of experiments involve nocturnal animals like mice and rats being used in the human working day when they should be asleep. And that's more than just an inconvenience to them. It's going to do more than make them feel bad. Every cell in every animal's body is like a ticking clock, and its functions vary with different times of day. So actually the experimental results that are obtained from animals at different times of day can vary hugely. And this is actually reflected in human medical prescribing now. There's a lot of thought about whether drugs will work better if people take them at particular times of day. So all these kinds of things aren't always recognised. The same will apply to genetically altered animals, and there's also a question mark there concerning whether effects that are seen are caused by the phenotype, by the genetically altered phenotype, or by the environment. And here's an example of a study on Alzheimer's disease using transgenic mice. So that long bit of jargon at the beginning there is the name of the strain of mice. Each strain has its own name that gives you information about how it was created. And these researchers used them and housed them in standard cages, like those little cages you see there. And they thought they just had a good standard model for Alzheimer's disease. But then they tried keeping them in larger cages with different floors and more for the animals to do. And they found it changed their science. So the plaques that appear in the brain with Alzheimer's disease actually appeared more quickly. So if you see the top right picture there, that's the mice in the enriched cage, and you can see there are more of those brown splodges in the brain. So that happened more quickly, but the cognitive function, the brain function, was lost more slowly. And the picture in the bottom right there shows how they performed in a test to test their cognitive power. And the ones in the enriched cage did better. So that actually taught them a lot about disease progression, and it goes to show that if you're just keeping animals in these standard cages, you don't always necessarily know what's causing the effects or what you're looking at. Despite this, though, genetically altered animal use is increasing hugely. This is the trend in the UK, and I think it's pretty much the same everywhere. And actually, one of the researchers who helped to develop the CRISPR-Cas technique has said she's been astounded at how quickly laboratories around the world had adopted the technology for a whole range of different reasons, but she'd avoided thinking too much about the ethics because she wanted to get on with the science. And I think this is happening more widely. So I just want to talk about how this is spilling out of the laboratory. And I was looking up the first ever genetically altered cow, and this cow came up. I'm not going to try and say that. I think it means dawn or morning. So that's her name in Finnish. And she lived 8 years, and she's stuffed in the University of Eastern Finland there. She was made from a cow egg from a slaughterhouse, and 12,000 micro-injections were performed. 19 calves came out of this, and just one success, which was her there. She was created to produce a hormone that stimulates red blood cell production in her milk. But this was never actually switched on because of issues with patent rights and health fears. So there she is, the first ever genetically altered cow, I think. So it's been proposed to, or in some countries this is happening already, to genetically alter farm animals for a variety of reasons. So increased yields, which I personally think should not happen, given that, according to the United Nations, 1/3 of all food produced globally every year is wasted, and 12% of all animal produce every year is wasted. So I think that needs to be sorted out as a matter of policy rather than altering animals. So-called welfare applications, organ sources for humans, not donors. An animal can't donate an organ. And also disease resistance. So just thinking about the so-called welfare applications, why I call it so-called, there are gene-edited cattle who are born without horns, which is presented as a welfare benefit, because obviously removing cow horns is extremely painful and distressing. But it is actually possible to keep cows together with horns. The thing is, it's expensive. So you can argue it's an animal welfare application because animals will have their horns removed, whatever, but ultimately it's done for economic reasons, I think. Also, the company that produced the polled cattle said they were precision bred, which is a phrase that you use a lot. But the United States Food and Drug Agency found that it had, they included antibiotic resistance genes from the CRISPR, so it wasn't quite as precise as they said. In the case of disease resistance, this is a slightly more complex case, I think, and here's an example from the UK. PRRS is a pig disease that causes breathing problems. It kills newborn piglets. It's very widespread. It does a lot of economic damage, so there's a very strong driver to fix the problem. The Roslin Institute has gene-edited pigs so that on the pig cell, the molecules that the virus used to attach onto are no longer present. So these pigs can't be marketed at the moment. They can't be released into the food chain. There are debates about whether it's a good idea to make animals more resistant or resilient to disease if they can still spread it. There are debates about the level of veterinary surveillance that animals can have. But the debate is very much ongoing both in the UK and in other European countries. including Finland. So when I was researching this, I saw that there's been a consultation, and I found that document there, which is very similar to discussions that we've been having in the UK. Our Ministry of Agriculture equivalent, DEFRA, underwent a public consultation in which they said it should be easier to produce gene-edited animals where they're not transgenic, so they haven't had genes from other species added. CRISPR-Cas has just been used to alter their genome, for example, to make them disease-resistant, like the pigs at Roslin. And they're saying this gene editing could have happened in nature, and so the regulations should be relaxed so that these animals can be produced and marketed. In our Queen's speech recently, it was announced there's going to be a genetic technology precision breeding bill, with the emphasis mainly on crops at the moment, but it says that animals will be included once it's demonstrated that it's safe to do so. But the public debate is very much ongoing, and I was disappointed. I felt that our government ministry had already made its mind up. And there's a real question, I think, as to who decides what's appropriate, who decides what should be done. So here's a quote from somebody from the biotech company that's working with the Roslin Institute. So she says, the benefits outweigh the risks, it won't affect the taste, and we should go ahead as rapidly as possible. So that's one opinion, but actually there should be a lot more debate than that. In the UK, we have the Nuffield Council on Bioethics, which has been doing a large body of work on gene editing farm animals. And this was one of their recommendations, that public authorities should support initiatives to explore public views about these matters, because the public are very concerned about this. Some people are concerned from an animal welfare standpoint. Other people are concerned as consumers about food labelling. Other people have environmental concerns, but the concerns are all there. So I just wanted to quickly mention other applications beyond farm animals. Here's an example, wild animals. We have a lot of problems with the American grey squirrel in the UK. Our native squirrels are red. The grey squirrel was introduced because somebody liked the idea of having grey squirrels in their garden, and they've just taken over almost the whole country, and you can see they do a lot of damage to trees. There's research ongoing to use gene editing coupled with what's called a gene drive, which really pushes through the gene edits much faster than would normally happen using normal sort of rules of inheritance. And what this gene edit does is it makes males sterile, and it reduces the number of females who are born as well. So this could be the solution. There are various solutions that have been suggested to reduce the number of grey squirrels, assuming they need to be reduced, which is another whole ethical debate. None of the solutions are particularly humane. Even if the gene editing does work, it will still be necessary to kill some squirrels as well. It won't work on its own. And also the impact on squirrel behaviour isn't properly understood. How will they behave? How aggressive will they be if the males are sterile and there are fewer females? And even if you remove a problem species from the environment, you're still perturbing the environment. It's not really known what's going to happen. Thinking about companion animals, here are a couple of examples. So the quotes on the left there are from a startup company that wanted to use gene editing to make dogs live longer. So they say people rely heavily on dogs. It would be good for companion dogs. It would be good for working dogs. But thinking back to the kinds of procedures that are done in order to generate gene-edited or genetically altered animals and the suffering and the wastage, would people who love dogs really want that? Similarly, it's been suggested that you could use gene editing to make cats hypoallergenic, so people with cat allergies could have them. Again, is that justifiable? The picture of the horse shows some horses who are actually created in Argentina by a research group that knocked out a gene called myostatin, which regulates muscle size. And so they produced an overly muscled horse that they said would conceivably be good at sport. So all of these applications, to me, really raise important issues when you think about who should decide. So here's another example, thinking about xenotransplantation, so genetically altered pigs whose organs can be used as human transplants, although somebody did pass away in the States recently who'd received a pig heart because of a pig virus. So this surgeon says it's better to give 1,000 people a 70% chance of survival with a pig heart than 100 people an 85% with a human heart, which makes sense, and I think a lot of people would probably agree with that. But some of the other examples I've shown you fall into much more of a grey area than that. They're not so straightforward. Should it be the people who have the facilities and the technology to do this who are ultimately deciding what should be done? So I think taking all of these considerations together, thinking about whether gene editing is different or not, I think it is. You know, I've sort of discussed the fact you can make multiple substantial, significant changes all at once. It's heritable, you know, it can be inherited like other ways of altering animals, but you're doing it instantly. It's specific and it's permanent. The fact that there are intellectual rights associated with these animals, you can patent these animals, also, I think, crosses a line. And there is just this lack of consensus about what it's ethically acceptable to do. So I think there's a big difference between gene editing inside the laboratory and outside the laboratory. Because inside the laboratory, as I've said, there's a legal framework, there's a system of licensing, there's a harm-benefit analysis, there's also a level of public accountability, because every establishment has an animal welfare body, which ideally would include a lay member, a member of the public. Now, this doesn't always happen, but where it does happen, at least you've got somebody there who's not directly involved with the research, who can be a bit more of an ethical compass and make people reflect on what they're doing. And the whole point with the laboratory animal legislation is that in each country, it's an enabling act. It enables people to do things to animals that they would not be allowed to do outside the laboratory. Whereas outside the laboratory, once this animal use isn't regulated by the national regulations on animal experiments, then it seems that this is when applications really run far ahead of what people understand and what people know is happening. So for those reasons, I think it's different. And another difference that often arises and that sometimes is discredited or discounted is issues around naturalness and animal integrity. So again, the Nuffield Council on Bioethics has done quite a lot of work on this, exploring what these terms like naturalness mean to people and how this actually means people should behave. And it's quite easy just to brush aside concerns about naturalness, to think that they're somehow sort of trivial or emotional. But I don't agree with that. And actually, when the Nuffield Council looked at this, they found it was what they called a placeholder for some real and legitimate concerns about human-animal interactions and about what it's all right to do to animals. There was a consultation in the UK about animals containing human material, because this was an area of ethical concern. So this could be animals who've had human genes put into them, for example, to create what's called a humanized model of disease. And there was a proper public consultation on this, which took the form of a series of workshops. And in the end, the public decided that they were unhappy about animals containing human genes if it made the animals appear human. They were unhappy about human genes being put into animals' brains, and they were unhappy about human genes being put into animals' germlines, so that they would be expressed in animals' semen or eggs. So those were carefully thought through, clearly expressed, and the legislation actually reflects that now. So the public were consulted, they did draw some lines, and everybody understands each other. This is ongoing at the moment with respect to gene editing farm animals. There's a wider public consultation underway, and it also includes vegetarians and vegans. So even if they don't eat meat, they're entitled to express a view and express their concerns about what's done to animals. Their view matters too. I think this kind of approach is really important. Everybody should be able to become involved. And just to sum up, really, sort of thinking of wider issues, I think the pandemic particularly has really made a lot of people focus on how humans and other animals interact, how we exploit other animals. particularly thinking about, encroaching on wild animal habitats. This has enabled a lot of viruses to cross from humans to animals. Also, farming practices have been the source of previous pandemics like Spanish flu. So personally, I am very uneasy about the thought of further mutating and changing animals to fit an environment rather than changing environments to fit animals. I think it's really time to stop pushing animals beyond their physiological limits, which is what's already happening. So just to conclude then, I think these are my main sort of take-home messages. I think genetic alteration and gene editing are more than just another tool to modify animals. I'd be really interested to see whether you agree or not. There's no right or wrong answer here. I think once they're applied outside a lab setting, there are additional ethical and animal welfare concerns that need to be addressed. I think everyone should be able to find clear information about the use of these technologies on all animals, and I think everybody should be able to add their voice to the debate and to play a role in decision making. So that was all I had to say. Thank you very much.